So our next speaker is, I cannot say your last name, Corinna Ehn.
Yeah.
She just finished her master's at University of Vienna, and she's just gonna follow on a PhD, and she's gonna talk us about pollination in Anthurium.
Yeah, I'm going to talk about the pollination and the nectar production in two co-flowering fermented fruit mimicking Anthurium species. Okay, so what is fruit mimicry? In fruit mimicry, the flowers are fooling a fruit-seeking insect by imitating fruits, for example, via the scent. And it exists possibly since the first fleshy fruits in the angiosperms occurred more than 80 million years ago. And the aim is to recruit the pollinators that either utilize the fruits as oviposition sites or as a fruit source. And usually, fermented fruit mimicry systems do not offer a reward. And the fruit model can be highly specialized, for example, when imitating a co-occurring species or a specific stage of ripeness. Fruit mimicry is known from gymnosperms and also a few families from angiosperms, also in Arum and Anthurium. Fermenting fruit mimicry in Anthurium is mentioned in the literature, but rarely studied in detail. And there are occasional sightings mentioned about insect visitors and also about liquid droplets occurring on the spadix. And in this study, I looked at two co-flowering species with fruit mimicry in Costa Rica. And my questions were, are there differences between the two species regarding the floral time and the chemical composition of the floral scent and also of the floral secretions? Which floral visitors are the pollinators, and are they also attracted by fermented fruits? And which compounds within the floral scents are responsible for attracting the pollinators? I did my study in the Golfo Dulce region in southwest Costa Rica in this red circle in La Gamba. It's a wet tropical lowland forest with more than 6,000 millimeter rainfall per year. So it's quite wet. My two species that I studied are Anthurium brownii and Anthurium clavigerum. They are both epiphytes, and they belong to the sections Spelodontium and Dactylophyllum. Anthurium brownii has an erect spadix. It's about 15 to 30 centimeters long, and it is dark purple to brownish. And Anthurium clavigerum has a pendant inflorescence. It can get quite huge, up to 70 centimeters, and it is purple in color. I looked at the thesis timing and the floral visitors. I did video recordings and direct observations, and every hour, day and night. I had 9 individuals of Anthurium brownii with 94 hours of video recordings and 40 hours of direct observations, and 7 individuals of Anthurium clavigerum with 37 hours of video recordings. I checked the sexual phase of the inflorescence, the presence and the absence of the floral scent, the pollen, the droplets, and the floral visitors. In addition, I observed the visitor behavior and if they get in contact with the reproductive parts or if they have pollen on their body. I did floral scent collection and analysis using the headspace sampling technique and later GCMS. And I did nectar collections using glass capillaries and analyzed them via UPLC and HPLCMS. And I looked at the amino acids and the sugars. I did bioassays using synthetic substances and also with fermented fruits. Coming to the antithesis of the two species, they have 2 sexual phases with no overlap. First, there is a female phase. It lasts about 10 days. And then is a male phase. It lasts about 20 days. And it is a repeating daily fluctuation of the aesthetic event at a specific part of the spadix. And it happens at daytime in Anthurium brownii, or at nighttime in Anthurium clavigerum. A little bit more in detail, in Anthurium brownii, the scent starts in the female phase in the early morning hours at 4 and lasts until the afternoon. And the stigmas produce droplets, which are visible here quite nicely. And the tepal secretions is sometimes visible as miniature droplets if you take a macrophotography, or it is visible as a film that is running down the spadix, mostly consisting of the stigma droplets. In the male phase, the scent starts at the same time, a bit earlier in the morning, and the anthers start to open at 1:30 at night. And here also the tepal secretion is sometimes visible as miniature droplets. In Anthurium clavigerum, the scent of the female phase starts in the evening and lasts until late at night. So it starts at 5 and lasts until 2 o'clock. And the stigmas have here also droplets, and also the tepal secretion is here clearly visible as small droplets. During the male phase, the scent is also in the evening hours, and the anthers open at nightfall. And also here in the male phase, there are distinct droplets on the tepals. Coming to the fluorescent chemistry, in Anthurium brownii we could detect 39 compounds, and the scent is dominated by ethyl acetate, acetone, and 2,3-butanediol. And you can describe it as sweet, alcoholic, overripe fruit. And in Anthurium clavigerum, we could detect 19 compounds, but it is mainly dominated by ethyl acetate with more than 70% and acetic acid. And I would describe it as fermented fruits vinegar-like. I looked at the sugar concentration, and the stigma secretions of both species have around 4% of sugar. And the tepal secretions of Anthurium clavigerum is 2-fold higher in the female phase, or 3-fold higher in the male tepal-derived secretions compared to the stigma secretions. And you can describe them as hexose-dominated or hexose-rich. A bit more in detail, the nectar mostly consists of fructose, glucose, and sucrose, and we could detect some other sugars and amino sugars, but only in smaller trace amounts. For the amino acids, we could detect in Anthurium clavigerum 11 out of 19 amino acids, in Anthurium brownii 7 different amino acids. Alanine, for example, is quite similar overall, the species and the secretion types. Or glycine is also quite high. What is interesting is, for example, in Anthurium brownii, serine is quite high, but not so high in Anthurium clavigerum. And Anthurium clavigerum has proline quite high in the tepal secretions, especially in the male tepal secretions. I could document more than 1,000 visitations by insects from 111 different species of different orders, mostly insects, and 79 species on Anthurium brownii and 47 on Anthurium clavigerum, and only 15 species are shared. And almost all species that visit both sexual phases and carry pollen or touch the stigmas while they are feeding on the pollen or on the stigmatic droplets. Sorry. A little bit more into detail, in Anthurium brownii, I found 10 families of Diptera, for example, these Drosophilid flies also touching the stigmas here, this Tephritide, Nearide, the Glyphotops, Sp and Rhopalomeride. 13 species of Hymenoptera, like this social wasp, Agelaya centralis, and stingless bees and ants. 7 species of Coleoptera from 5 families, this fruit beetle, Hoplobugua litorata, or the sap beetles, Snititulide, covered also in pollen, and also this beetle. I also marked several individuals of the fruit beetle Hoplopyga litorata, and I could show a movement from the male to female inflorescences. 11 species of butterfly, all nymphalidae, like this Hermaptychia hermes, feeding on the droplets and touching the stigmas, and 1 unidentified moth. These are 6 other butterflies that I encountered. All are known to be fruit feeding. Here's a short video. There's Nika Flavilla in the beginning, scared away by Hamadryas feronia. Now he's landing and probing with the proboscis. And butterflies stay up to 20 minutes most of the time. This is Calicora lyca on a male inflorescence, also probing with the proboscis and moving downwards towards the base. And Nika flavilla. Consul Fabius, I'm sorry. Also probing, and he has to share the spadix with a lot of ants, as you can see. Okay. In Anthurium clavigerum, I encountered 7 species, or about 7 species of moths, mainly Erebide, like CF thaleviridans, or this huge one, Ascalapha odorata. Or here in this picture, Latest Budea at the base and Ascalapha odorata at the bottom, sitting down while feeding. And here in the video, you can see another Erebide that is not identified, flying up and down the spadix, the tip that is synthetic at the time, and drinking the liquid droplets. Here is a calmage, and I cannot go on. Okay, yeah. If you go into detail, you can see this Erebide has pollen on the proboscis and on the legs. Oh, no. Yeah, I also encountered katydids. Next, please. Yeah. And this pollen on the tarsi we could detect. I had cockroaches on female and male phase inflorescences, nymphs and adult ones, several species probably. I did bioassays with fruit baits, and I could detect most of the visitors that are visiting the plants also come to the fruit baits, like Letis budea or this fly Willis thorniella. I did bioassays using synthetic substances. These 6 ones, I did different mixtures. I also tested them pure against the negative control, and I tested also mixtures against mixtures. Overall, I attracted just Drosophilid flies. I used the sentinel traps for this. And overall, you can say they attracted more or less the same number of species. I could not say that one mix is better than another one. Maybe I can say that the mix containing 2,3-butanedione is maybe a little bit more attractive to them, but I can't. But the mixture is just important. Next, please. Okay. So I have here 2 sympatric flowering species with distinct temporal niches. In Anthurium clavigerum, the pollinator attraction is in the evening and it lasts past midnight. And in Anthurium brownii, the pollinator attraction is in the early morning hours till the afternoon. I have here a generalistic system for fruit feeding insects. And Anthurium clavigerum is most probably pollinated by moths, maybe cockroaches, katydid not so really. And Anthurium brownii, I think it is you can say that Drosophilid flies and other fruit-feeding flies, butterflies that are fruit-feeding, and these fruit-feeding beetles can act as pollinators. Comparing this with other fruit mimicry systems, you can see we have a quite generalistic system compared to this because, for example, other mimicry systems are, for example, pollinated by families or from a genus or even just from one fly species. And the scents are similar, so in bold is indicated the scents that we share with these systems. For example, ethyl acetate or acetylene or 2,3-butanediol diacetate. They are found in different mimicry systems as well, and all the chemical compounds are known from fermented fruits. Coming to the nectar, in Anthurium, this is still a controversial and debated thing. In 1905, Engler first mentioned that stigma droplets contain sugar, and 1930 Daumann described tepal secretions with sugar. And 50 years later, Tom Croat showed that Anthurium cyperti contains 8% sugar in the stigma, consisting of sucrose, fructose, and glucose. And 2021, Paiva et al. Showed sugary tepal secretions derived from nectaries. And several studies showed that there are visitors coming to the inflorescences feeding on the secretions. And we show that the stigma and tepal secretions have several sugars and amino acids, which are consumed by the pollinators. And there is now clear evidence that Anthurium has nectar. To compare our nectar with other nectars from other plant families, Anthurium has, our two species have a mean of about 4 to 13%. Compared to other floral nectars with a mean of 20-25%, we are more in the lower range. But nectar sugar concentration can vary widely, also between and within species and families, and they all strongly can depend on the pollination system. For example, butterfly pollination systems are known to have sometimes 13% of sugars, or ant with 5%. And comparing this to tropical overripe fruits, they have a mean of 1 to 4%. So we are in line with these tropical overripe fruits. And what is interesting to say is that sugars decrease during the fermentation process in fruits. And our amino acid content lies within the range of nectars of other flowering plants. And there are similarities also with fermented fruits. And in fermented fruits, the amino acids increase during the fermentation. Next, please. Yeah. To conclude this all, we have two generalistic and rewarding fruit mimicry systems discovered, and these pollination systems seem to be maybe an important food supply for several rainforest insects which depend on fruits otherwise, and such interactions might be found all over the neotropics. Sorry, yeah, thank you.
Thank you. We have time for one or two questions.
Hi, Corinna.
Hi.
Really beautiful work. Thanks for showing it. Am I right to think then that you think that they are mimicking fruit fermentation generally rather than specific plants? They seem to be attracting lots of different types of insects. So do those insects all share some particular types of fruit that they eat?
Mm-hmm.
And yeah, I guess that's the question. It's a little bit hard to see this on the odorant side, since basically your headspace is going to be completely dominated, right, by just general fermentation byproducts like acetic acid or whatever.
Yeah. Yeah, I assume they are just imitating fermenting fruit. I don't know. I would need to collect fermented fruits in the area and compare the scents and maybe also the sugar composition. so that I can say, okay, they are mimicking, for example, this specific fruit and this specific stage of ripeness. But it is difficult because the composition of the scent, for example, changes over the ripening stages. So I would need to cover a lot of ripening stages. Those are a lot of species. But I think they are just more or less, in general, mimicking some fermenting fruit.
Another question?
It seemed that the phases for fragrance and droplets were a little bit out of sync or non-overlapping. Is that right?
What? I
When
didn't get the
The
question.
timing of fragrance and the timing of the droplets was different?
No, also when they are enthetic, when they are enthetic, they produce the scent, and in this time they also produce the droplets. But the droplets, for especially the tapular secretions, start a bit earlier. So
Why do you think that the stigma droplets are weaker than the tepal droplets?
maybe because they are more like these germination drops in gymnosperms, maybe to get the pollen to the stigmas, but
Right. And did
I
you
don't know.
observe any predators?
No.
Thank you very much, Corinna.
Thank you.